From Microbe TV, this is TWiN, This Week in Neuroscience, episode 62, recorded on July 21st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York, Tim Chung.
Hello. Hey, Vincent. Hello. Good to be back. It's just the two of us today because Vivian is away.
Yeah. I think we did a two on not too long ago also, you and me, right?
My memory does not extend that far back.
All right.
Oh, no. Quite possibly.
So you said you were in Vietnam recently.
Oh, no,
right?
You're right. It was you presenting, I believe. Yes, I was in, so we were just talking about before the show. I was in Vietnam on holiday maybe a couple of months back.
It's very beautiful, isn't it?
It's absolutely lovely. They have like wonderful kind of karst mountain next to the coast. So Vietnam is basically a long strip of coast where people live. And right next to it is like high mountains and rainforest. And then with a lot of old ruins, kind of Angkor Wat style ruins from old Tamil civilization.
I'd like to go.
It's wonderful. It's getting kind of rapidly developed right now. So, and there are just mopeds everywhere. It's unbelievable.
For some reason, I get a lot of Instagram posts about Vietnam, and they get a lot of flooding, and they show cars being washed away, mopeds being washed away. You know? And then recently it was the lychee nut season, and all these mopeds with huge bundles of lychee nuts on their backs crowding the streets.
Oh, wow. Okay. That's amazing.
But supposedly the food is really interesting and it's got beautiful scenery.
Food is amazing because it's it's an old French colony, so it has a lot of French influence, but it also obviously has kind of native Vietnamese pho noodles are very famous as well. So it's a very nice blend. Family had a really good time in Vietnam, had some really wonderful food. But that actually relates to what we're going to talk about today because
it does. It
does talk about, I hope people listening aren't having the lunch right now because the lunch might come up. But on one of my last days in Vietnam, I actually had suffering from pretty bad food poisoning. So TWiF listener would know of the expression called two buckets disease. I would let your imagination fill in what that means. But it was highly unpleasant. And I think it was because my dad ordered like a dodgy takeaway from a random Vietnamese, maybe home-cooked meal. And after that, I basically I don't think I'm going to eat Vietnamese food for a year just because of that. But so that's what we're going to talk about today is basically how we learn to avoid certain food after it's made us feel sick. And I think, I don't know if anyone has had the problem of getting food poisoning before, but usually after you suffer from a bout of food poisoning, first thing you think about is what did it? Like, what food is it that I ate? So you can try to like reason using your memory, kind of like logically, oh, I ate this food, it must be this. But often, next time when you're faced with the same food again, let's say you go back to the same restaurant, you just feel an urge to avoid the same food. Or maybe if someone else brings in a food, so for example, out of Vietnam, I was supposed to meet a group of friends for dinner, and they selected a Vietnamese restaurant, and we had to call them and said, please, can we change? Because I cannot stomach any Vietnamese food.
Yeah, that's funny.
So you would avoid the same food that just caused you the sickness. And what's very interesting from a neuroscience point of view is that it actually takes a while between when you eat the food. Usually it takes a while between when you eat the food and the sickness that it causes. So for example, when I got sick in Vietnam, I believe it took maybe an hour or two after dinner when I was like, oh no, I ate something bad. And that's actually quite interesting because in a lot of animal study literature, we have done quite a lot of work on so-called associative learning. So remember, for you to learn to avoid the same food again, you have to associate the original food's flavor, the smell, the taste, with the nausea that comes afterwards. And in a lot of animal studies, we have done similar kind of experiment of associating thing one with an outcome. For example, you can associate the pairing of a tone in a chamber with a shock. And people have done that with mice and rats. And even I, when I was a young wee undergrad in England, I remember running experiments where I had to I'd train rats to press a lever, and after a certain delay, after the lever press, a food would come out in a hopper and the rat would go eat it. And these rats would absolutely learn to lever press. If there's no delay between the lever press and the food, they would just press the lever like mad. Mm-hmm. But even if you put in a delay of maybe about 10 seconds, mm-hmm. The learning, the response rate completely plummet. And in the literature, I think they found that if you up the delay between the lever press and the outcome, the food, to 60 seconds, then the rats just don't learn. And I've heard I've never run the experiment, but I've heard mice are even more sensitive to this kind of delay. So like 60 seconds to learn stimulus and its outcome actually is quite tough for animals.
Humans can probably do that, right?
Humans might be able to do that. So, for example, imagine this very simple experiment of you coming in New York City, you go to a traffic light and there's a button. Mm-hmm. And the button, if you press it, maybe it would make the light change color to allow you to cross. But let's say there's no feedback that you actually press the button and it did anything. So you press a button and the light doesn't change for a minute, and then it changed. Do you know if the button is working? Like, try to figure that out. It's actually it's quite hard
because the lights change automatically.
Especially if the light changes automatically anyway. Yeah. So if it's quite hard to figure out if the button is doing anything, if there's a delay between.
I think New York, most of them are broken. Anyway.
Exactly. Pretty sure it doesn't do anything. It's the same with our elevator here in the building. Like, there's a closed door button that absolutely does not do anything. And like, you can tell who is new to the building because they would try to press that button. So there are all these, in the wild, in nature, in real life, there's a lot of these association that just for us to kind of survive, we have to learn. And it can be quite hard because of the delay. So in like all these psychology experiments that we subject the animals to do, they can't even bridge a delay of 60 seconds. But with all this, with this food sickness association, the delay can be 30 minutes to 2 hours. So it is quite a mystery, actually, in the field of how all of us can do it. So that brings us to today's paper, which is from the lab of Ilana Witten. And the first author is Christopher Zimmerman, and there's a long list of authors, so I won't go into all of them. I'll just say the first author is Zimmerman, and the last author is Witten. And these are everyone's from Princeton, makes my life very easy.
Yeah, and also the affiliations are right on the front page there instead of at the end of the article, which sometimes happens, right? Yeah,
makes it super easy. So it's Princeton University, and also Howard Hughes Medical Institute, HHMI. And it's published in Nature, and I believe it's open access. If not, I'll send a link to Vincent to make it open access. But the article published in Nature, I think, just a month ago, actually, this year, 2025, is called A Neuromechanism for Learning from Delayed post-ingestive feedback. All right, so again, that's mumbo jumbo, but would explain a little bit more in the course of today. But post-ingestive feedback just mean basically the sickness, the nausea or the illness that you feel after you eat a dodgy meal.
But you also, if there are some times when you eat bad food and you taste it right away, right within seconds.
Exactly.
And you don't eat anymore, and then maybe you won't do it again next time. Also.
Exactly. So that's very, very good point, Vincent. So there are a couple of things that would make you not want to eat something. One is the taste. So for example, most animals and also most children and some adults will not eat bitter stuff. For example, things like coffee is probably an acquired taste. And that has something to do with probably has something to do with the bitter sensor in your tongue projecting to the insular cortex, the gustatory cortex. And then there are studies by most prominently Charles Zucker in Colombia, actually, suggesting that there's these things called labeled line projection, where all the bitterness receptor in your tongue projects to this place in the taste cortex, the gustatory cortex, and they all project to this place called the amygdala that we'll talk about. And maybe the amygdala is a kind of an aversion center for the bitter taste, so that you taste it and you're like, ugh, this is not good, I don't want to eat it. And these are maybe innate kind of preference that you have for things that might be toxic, like a lot of, poison out there, it tastes bitter because of that. And that's like an evolutionary drive to avoid those food. But today, we're actually going to learn about things that might initially taste really good and that mice would love to drink it, but you learn that it is bad, so you avoid it. So let's set up the paradigm of how they're going to get at this problem of kind of a super long delay between the thing that you taste and the sickness that comes afterwards. So they come up with this paradigm where the name is called conditioned flavor aversion, CFA paradigm. And what happens in this paradigm is you get a mouse and you give it a flavor that it has never tasted before. And specifically in this case, they use this thing called sweetened grape flavored Kool-Aid.
No mouse has ever had Kool-Aid, right?
I've been in this country for a very long time, and I have never had Kool-Aid.
You're not missing anything. You're not missing anything.
To be honest, the name itself already sounds like it's poisonous. But apparently, mice would like it. If it doesn't, mice would drink it if it doesn't lead to any sickness.
Tim, many years ago, this cult in some country following Jim Jones, he used to give them Kool-Aid with poison in it, right?
It's in Guyana.
So now that Guyana, now we all say you drank the Kool-Aid, right?
Yeah.
Because you just, if you're working in a company and you listen to the head and you say, yeah, you've drunk the Kool-Aid.
And I think, like, horrifically in the Jonestown massacre, he put cyanide in the Kool-Aid, which is a poison. And if you survive the cyanide, you probably would avoid the Kool-Aid in the future.
That's right.
Like the mouse does. But today, they didn't pair the Kool-Aid with cyanide, thankfully. What they did instead is they exposed these mice to the Kool-Aid, which is novel. They've never tasted it before. And then 30 minutes later, they injected the mice with this thing called lithium chloride. So not sodium chloride, which is basically saline, but lithium chloride. And lithium chloride triggers in mice and presumably in human a deep nausea. So they call it malaise. So you just basically don't feel good if you get given lithium chloride. And then 2 days after this single pairing, so it's a one-shot learning. They just do it once. 2 days later, they gave the same mice a choice between water in one cup and the Kool-Aid flavor in another cup. This is called the retrieval test. And what you see is that the mice that are exposed to the lithium chloride malaise, they much prefer the water and they basically reject the novel flavor, the Kool-Aid. But in a different group of mice, this is the familiar group, they actually pre-expose these mice to 4 daily exposure of Kool-Aid first. So these mice have learned that the Kool-Aid, they don't know that it's called Kool-Aid and has a weird name. They just know that it tastes kind of like grape, maybe some artificial flavoring, who knows. But they drank it after 4 days. They're like, okay, it's safe. And then after 4 days, if you now present this now familiar flavor to the mouse, and then 30 minutes, do the same lithium chloride injection to trigger malaise. So the malaise would be the same. The mouse would still feel bad. But then 2 days later, you test the mouse again on the retrieval test. The mice now no longer reject the Kool-Aid. They would actually drink the Kool-Aid as much as they drink water. if not actually a little bit more, because presumably Kool-Aid tastes good. So this tells us that, first of all, if the flavor is novel, then if you pair it with lithium chloride or malaise, 30 minutes later, the mouse would trigger this conditioned flavor aversion. It would reject it in the future. But this can be overcome if the mouse has been pre-exposed to the same flavor, so that the flavor now becomes familiar. So using this kind of behavioral paradigm, the authors went in and asked a question, all right, where in the brain might this whole system work? Where is the memory stored? How does a mouse learn? Well, first of all, how does a mouse learn initially when you pair this Kool-Aid and with lithium chloride? And then later on, when the mouse has to retrieve the aversive memory when you give the mouse Kool-Aid again, without any lithium chloride, just a Kool-Aid, how does it retrieve the memory so that it doesn't drink it anymore? So to do that, the researchers basically did this thing called brain-wide FOSS imaging. So FOSS, we've talked about FOSS quite a lot on this podcast already, and it's basically, FOSS is an immediate early gene. It's a protein that is made by a neuron that has recently been firing. So if a neuron has been recently firing, then within 30 minutes to an hour, it would start making this FOSS protein. And this FOSS protein is made, and then quite quickly afterwards it degrades. So it's kind of like a pulse that you can chase for when a neuron has recently been active. So this kind of substitutes as a fMRI scan for mice. You can't put a mouse in an fMRI scan. They won't sit still. So the next best thing is using this, you do something that you want, and then very soon after, you euthanize the mouse and you look at the brain and you see where these FOSS are expressed in the brain.
And you can't anesthetize the mouse for an MRI, no?
Well, yes, you can. So if you anesthetize the mouse, then the mouse won't move around and won't give you a blurry fMRI image. The downside is you can't make the mouse do things. So the whole point of fMRI is that the mouse is doing some behavior, is performing some function, and then you look at which bit of the brain is changed. So you either have the mouse sit still and anesthetize and get a clear image, or you have the mouse do something you want and you get a blurry image. So you can't quite get the sweet spot in the middle, unlike in human. They actually have the same problem with scanning babies if you want to look at how babies' brain work. Anyway, so instead of doing fMRI on the mouse, they did this brain-wide FOSS imaging. So they actually had three. So importantly, they divided the conditioned flavor aversion task that we just talked about into 3 time points. So this is quite important because the 3 time points are the 3 phases we think that the mouse as a system needs to go through to learn this task. The first time point is called the consumption time point, where you just present the mouse with the novel flavor at the beginning. Or they actually have 2 groups. Either the mouse got the novel flavor, so they saw the Kool-Aid for the very first time, or it's a familiar flavor, so the mouse saw the Kool-Aid for the 4th time. So this is just consumption. No lithium chloride given, no malaise. The mouse is just like tasting, this is novel. Maybe I want to give it a second lick. So that's the first phase. The second phase is the malaise phase. And this is after you give the mouse the flavor, and then 30 minutes later, you give it a lithium chloride injection to trigger the sickness. So in this malaise phase, what you're looking at is maybe what does the feeling of nausea, how does that activate the brain? And also, how does any brain that got activated by the flavor, maybe it would also have to respond to this sickness and therefore get activated. And then the final phase that is important in this paradigm is the retrieval phase. So this is, you have trained the mouse on this, you paired the mouse in this flavor and lithium chloride sickness pairing already. You wait 2 days, and then you re-expose the mouse to the same flavor. And if the previously paired flavor was novel, the mouse would reject the flavor. But if the previously paired flavor is familiar, then the mouse would actually still drink the flavor. So the question is, when you present this on the retrieval session, you give the same flavor again, what goes on with the brain to explain these kind of differences. So in this phase, you're looking at the recall of the memory, whereas before, especially during the malaise phase, you're maybe you're looking at the formation of the memory. So what did they find? They found that for the consumption time point, so at the very beginning, the very first exposure of the novel flavor versus if it's familiar, if it's a novel flavor, then the bits of the brain that got activated as assayed by this FOSS is a lot of sensory cortex. So as Vincent was saying earlier, as you drink a new flavor, maybe your taste cortex, your gustatory cortex, also known as the insular cortex, it lights up, and that's what they found. And also the piriform cortex, which is the smell cortex, it also lights up. And another area that lights up is this area that we also talked a little bit about called the amygdala. So the amygdala is very interesting. First of all, it actually receives a lot of input from both the taste cortex and the smell cortex. Secondly, it also has been previously known to be very important in association, of learning all these kind of Pavlovian conditioning, pairing. If you train a rodent to pair a tone with a shock, the rodent would learn it and freeze to kind of future tone. If you just play a tone, the rat would freeze. But if you ablate the amygdala, the rat would no longer freeze. So it tells you that amygdala is very important for the formation and also the expression of this freezing behavior. So it's important in learning. And people also found that the amygdala, people previously also found that the amygdala is important for this kind of nausea, malaise, learned aversion. But conversely, for the mice that are tasting the Kool-Aid flavor for the fourth time, so it's now a familiar flavor, what they found is a completely different network of brain that are activated, and they are what people have colloquially called it the limbic region. We won't get into the terminology here, but those are bits of the brain that are normally associated with kind of emotional processing, including things like the nucleus accumbens that people sometimes think of as the reward center. Also the hippocampus, very important for memory formation. But what's interesting that no one has reported before is this area called the lateral septum, which is involved in a lot of kind of emotional processing and also a lot of innate behavior processing that we won't talk about it too much because frankly I'm not an expert. But because this area, lateral septum, has never been reported before to be important for learning that a flavor is familiar and is, you can imagine it being a safe flavor to eat. And it's actually, let's quickly talk about this. It's really important for rodents to learn whether a flavor is safe to eat or not, whether it's toxic, because they actually have as far as I can tell, they don't have a vomiting reflex. So once they eat something, that's it. If it makes them sick, they can't throw up again. They have to just sit there and let it pass through. So it's very important for them to learn that a flavor is actually safe and they can go back and eat it. And it seems like once they have learned a flavor is safe, the lateral septum is activated. And these researchers went in, took a deeper dive, and they asked a question, well, if the lateral septum is activated when a food is safe, if we activate this lateral septum, as the mouse is learning this pairing of novel Kool-Aid with lithium chloride that triggers nausea, can we block it if we just artificially activate the lateral septum? Can we trick the mouse to think that this novel flavor is actually a familiar one and therefore is safe and therefore is probably not the thing that's causing the nausea? And they used this technique called chemogenetics, which is basically you express a designer receptor in the lateral septum that would recognize this designer drug that you inject and would be activated by it. And they show that if you artificially activate the lateral septum, you can indeed block the formation of this conditioned flavor aversion. So the mouse would no longer reject the previously novel Kool-Aid that in normal circumstances it would reject. But presumably when you activate the lateral septum, the mouse thinks it's safe. And also what is also interesting is that if you give a novel flavor, the researchers found that the the amygdala network has a lot of FOS, so it is actually activated. But if you, at the same time, activate the lateral septum when giving the mouse the novel flavor, the amygdala FOS, the amygdala activation gets suppressed. It's once again suggesting that maybe it is you're tricking the brain into thinking that the novel flavor is actually familiar and perhaps safe. So quickly to summarize, when you give the mouse a novel flavor, the amygdala seems to be of strong contention for the thing that gets activated. Whereas if it's safe, the lateral septum seems to suppress the amygdala, tell the mouse it's okay to eat. So the next time point is the malaise time point. When you give mouse a bout of sickness and pair that with a novel flavor, what happens to the brain? So the main thing that the main region that gets activated in the brain is this region called the parabrachial nucleus, which is all the way back in the brainstem where a lot of these kind of innate processes happens.
It's far from the amygdala, right?
It's very far from the amygdala. It is kind of all the way at the back of your brain near the cerebellum. It actually receives input from this place called area prostrema, also known as the vomit center. But it is known to send projection all the way to amygdala. So people have previously shown that. So it is a kind of the usual suspect that you expect to light up, and indeed it does. But a second network that lights up is once again the amygdala. So during this malaise time point, both the kind of the malaise sensing region, the parabrachial nucleus, and the amygdala lights up. And finally, for the retrieval time points, the region that lights, the network that lights up is again the amygdala network. So, and the authors kind of show this using hierarchical clustering. We won't get into the details, but they did some fancy analysis of the whole brain so that you control for the fact that you have, 200 brain regions and you don't want to get any false positives. So there's a lot of modeling going on, but they show that in the retrieval phase, and again, remember, during retrieval, the mouse would reject the flavor only when it was previously novel. And only if with a novel flavor did you get a strong activation of amygdala. So it all suggests that amygdala is the prime suspect for some to be involved in this kind of learning because it is activated in all three time points. The initial novel presentation of the flavor, during the malaise pairing of the flavor, and during the retrieval of the memory of the flavor at the end. And this is actually quite an interesting finding because, first of all, the fact that the novel flavor still activated the amygdala during the malaise time point suggests that because the malaise time point is like 30 minutes, it was given 30 minutes after the initial exposure to the novel flavor, it suggests that maybe something is going on in the amygdala to bridge that gap of the 30 minutes. So maybe this is the place where, so there's this concept in psychology called eligibility trace or memory trace. So for example, if I press the button for the traffic light in a crosswalk in New York, I have to remember that I just pressed this button so that later when the light change, I can assign to the button press as the cause of the light change and not, some guy next to me talking on the phone cause the light to change. So there are all these kind of, psychologists have kind of theorized that there are all these memory traces that happens in the brain every time you do an action or you are exposed to a stimulus. There are these memory traces in the brain that decays as a function of time, so that the closer you are to the occurrence of the stimulus, the stronger the memory. But as time goes on, the trace decays away, so that it's harder to learn about the pairing as time goes on. It's
not just memory, is it? It just goes away?
We don't know. So this eligibility memory trace thing is a hypothesized thing that might be happening in the brain to try to explain how memory is formed and why it is sensitive to time. But it is really hard to identify. And I think one of the contribution of this paper is that it tries to answer that in this case. So we'll get in a little bit into it during the last part of the paper of where this trace actually is kind of physically. But this FOSS kind of brain kind of fMRI analog suggests that this memory trace might still be there after 30 minutes, and it's in the amygdala. And finally, another thing that's important is that because the amygdala is once again reactivated during retrieval, it suggests to the researchers that maybe the amygdala is the site for both the storage of the memory and also the recall of the memory. So that is, so they identified the amygdala being a crucial player probably in the formation and the storage of this process. The next thing that they try to tackle is how does the brain tell the amygdala that it is feeling sick? So remember, the brain region that got activated by the malaise, by the lithium chloride, is the parabrachial nucleus, all the way in the brainstem, close to the vomit center. So they want to, what the researchers next did was they want to show that the parabrachial nucleus is activated by lithium chloride, by the sensation of malaise. Quick sidetrack, the parabrachial nucleus contains several different types of neurons. And one of these types of neurons have been previously found to be very important for kind of the feeling of nausea. And there are neurons that express this peptide called calcitonin gene-related peptide, also known as CGRP. So this is just an alphabet soup peptide that we're going to use for today's purpose. It basically identifies this group of neuron as a different subtype of neurons inside this brain region. So it's a marker for this type of neuron. But people have previously been found that this particular subtype of neurons, first of all, project to the amygdala, and second of all, might be important for the feeling of nausea, and especially might partake in the formation of this nausea memory that we're looking at today. So what the researchers did was they did this technique called fiber photometry, where they expressed a calcium dye in these CGRP, calcitonin peptide. I'm going to shorten it to calcitonin neuron, even though they don't make calcitonin, they make a peptide related to calcitonin. The CGRP neurons, they express a calcium sensor so that these neurons would glow when they have calcium in them. And usually neurons have calcium in them when they fire, calcium enter the cells. So what they saw is that as soon after they inject lithium chloride, these CGRP neurons in the parabrachial nucleus, they start glowing, suggesting that they start firing quite strongly after nausea. So that's great. That kind of squares with a FOSS finding. The next thing that they showed is that these calcitonin-related peptide neurons are actually projecting monosynaptically. So there's a direct connection to the amygdala. So they did it via two ways. First way is in both ways, they express this thing called channel rhodopsin in these calcitonin neurons. So channel rhodopsin is basically optogenetics, where you shine a light and the light opens these channels in these neurons and causes the cell to fire. So what they did was they recorded from the amygdala, and then they shine a light to make calcitonin neurons fire, and they were able to observe basically what appears to be glutamate currents. So it suggests that the calcitonin neurons are directly releasing glutamate onto the amygdala neurons and causing these glutamate currents to go through. And these glutamate currents would normally make the cell fire. And indeed, they also did it. So previously did that in slice, using slice electrophysiology. They also did the same thing in vivo in a living, breathing mouse by shining a light in the parabrachial nucleus to make those cells fire, and then recording in the amygdala. And they show that whenever they shine the laser into the make the calcitonin neurons fire, in very time-locked manner, the amygdala also fires. So it tells you there's a direct connection between the calcitonin neurons in the parabrachial nucleus, the kind of like the vomity, nausea sensing center, and the amygdala.
Those do not respond to the initial flavor. They respond to the malaise, right?
Exactly, yep. They respond. They are the neurons that responded to the sickness. And so these are kind of correlative studies showing that there's a connection and that, whenever the mouse feels sick, these cells fire. Now they want to ask, are they causal? Can these cells, if we artificially activate them using optogenetics, can they trigger learn aversion to this Kool-Aid novel flavor? So that's exactly what they show. They show that if you give a different group of mice, you give them this novel flavor, 30 minutes later, you artificially, using optogenetics, stimulate these calcitonin related peptide neurons. Then 2 days later, you test them, the mice would reject the novel flavor, which is great. They show that activating these neurons can mimic nausea, even though the mouse didn't get lithium chloride. So the mouse, presumably, the stomach is fine. It's not having any diarrhea, perhaps, but it still felt nauseous. Conversely, if you use a different type of optogenetics to inhibit these neurons, and so in this experiment, they once again have the novel flavor. 30 minutes later, they actually went ahead and gave lithium chloride. So in the control mice, they subsequently rejected the novel flavor, the Kool-Aid. But in the experimental group, they used optogenetics to inhibit the calcitonin peptide neurons. And when you inhibit these cells, even though the mouse was supposed to be feeling sick, you can block the aversion. Not completely, you can partially block it because, as it turns out, the calcitonin, there are other bits of the brain that might also partake in this memory formation. But they show
So this parabrachial nucleus, you can send two different kinds of signals. Well, actually, you send a signal which will block the aversion, and then if you prevent a signal from going, it causes aversion. Is that right?
The other way around. So it's the same signal. So calcitonin related calcitonin peptide neurons. They are just activated by malaise, by lithium chloride, and they directly activate amygdala.
To the amygdala,
talk to amygdala, right. And you can basically mimic it artificially using a stimulating optogenetics. Right. But there's a different type of optogenetics, which is the inhibiting optogenetics. And if you inhibit these neurons, then you can, even if you give lithium chloride, you can block the nausea memory formation.
So inhibit means there's no neurotransmission, right?
Exactly. You block the release of neurotransmitter from these neurons. So by doing so, the researchers show that the calcitonin related peptide neurons are both necessary and sufficient to trigger this learned flavored aversion, which is a very strong kind of gold standard way of showing that this brain region is involved. Next, they actually asked the question, if this parabrachial region with this calcitonin related peptide neurons, if they're involved, then we should be able to trigger a similar kind of brain-wide FOS activation just by artificially stimulating this group of neurons. Mm-hmm. So once again, they repeated the experiment where they presented the mouse with either a novel flavor, the novel Kool-Aid, or the familiar Kool-Aid. And then 30 minutes later, they did this artificial optogenetic stimulation of these calcitonin-related peptide neurons. And what they found is, indeed, they can actually replicate the activation of the amygdala using FOS. So it suggests that this artificial stimulation alone can basically trigger something that looks a bit like the memory formation step during what we talked about previously, during the malaise step, the similar activation inside the amygdala.
So what we know is that the activation will have that effect on the amygdala, but we don't know the nature of that activation, right?
We don't know the nature of the activation, and we also don't know. So remember, the amygdala was activated when you first, during the initial exposure to the novel flavor, when you first go in and try a food, and you maybe similar to babies, they're a little bit suspicious about new food. Same with mice. When you first give the novel exposure, the amygdala lights up with FOS, and then when you give it kind of the malaise sensation 30 minutes later, the amygdala also lights up. What we don't know, though, is it the same? We know the same region lights up, but is it the same neurons?
Yeah.
So that is what the researchers address for the next set of experiments. So the next set of experiments are very interesting and it's quite amazing that they did it. It's a lot of work went into it. What the researchers aim to do is they have 3 different hypotheses that they are proposing to try to explain this kind of formation of this very delayed memory. The first hypothesis is that when you're initially exposed to the novel Kool-Aid, the certain groups of neurons in the amygdala start firing, and they are the neurons responsible for coding this novel flavor. And they remain firing for the entire 30 minutes of the delay, such that after 30 minutes, when the calcitonin peptide-related neurons, they get triggered by the malaise, the calcitonin neurons are able to talk to the same amygdala neuron they're already firing, and this forms some sort of plasticity. So plasticity is what neuroscientists use to kind of like a general broad term to suggest there are some synaptic changes that might kind of underlie a memory formation. So maybe the memory trace is formed by these neurons being consistently and persistently firing during the delay, and that's how they bridge it. Now, the second hypothesis is that during the delay, these neurons actually don't fire anymore. So there's an initial firing when you're exposed to the novel stimulus, the novel flavor. But during the delay, these neurons don't fire. And somehow, for reasons that we don't quite know, when you're exposed to the malaise, so when you're exposed to the stimulation from the calcitonin-related peptide neurons, that re-triggers the same neurons to restart firing again. And it is this reactivation that causes the memory to be formed. And the third hypothesis is that, once again, during the delay, there's no longer any firing. And when you present the malaise later on, 30 minutes later, a new group of neurons pop up. And this new group of neurons are the malaise sensing neurons in the amygdala. And they get incorporated into the original novelty flavor population, so that you have a big giant population of neurons that encodes that this flavor is both novel and it is nasty and it causes you sickness. So this is now, hypothesis
if you give the mice one flavor and then that would activate a certain set of neurons, would a different flavor activate a different set?
They did not test that. So that's a very good, interesting question. They did not test that in this experiment. They only tested grape-flavored Kool-Aid and water. So it's not. So water presumably has no flavor. Right. Although if you're in New York, it can have some flavor.
Yeah, there's always some flavor to water, right?
Depending on how brown your water is. Down in Princeton, they might have kind of fancier water down there. But at least according to in this present experiment where they compared Kool-Aid flavor versus water. So this is now. The researchers now have inserted these very, very kind of cutting-edge technology, electrophysiology probes, recording electrodes, called neuropixels, into the amygdala. So these are basically recording electrodes that, so once upon a time, you can only record from one neuron at a time. This is only like maybe 70 years ago, you can record one neuron at a time. And then that turned to maybe 4 neurons, maybe 10 neurons at a time, when you can insert like 4 wires into the brain called tetrodes. But nowadays, people have come up with ways of putting in a single recording electrodes, having maybe between 100 and 300 different recording sites. So we can record hundreds and maybe even thousands of neurons at a time. So technology has improved, has advanced unbelievably. So this is all very good. So the researchers use these neuropixel recording to record from, I think on average per mouse, 140 neurons in the amygdala. And what they found was that during the initial consumption phase, where you present the mouse with a novel flavor versus water, and water is presumably familiar, what they found is that they can, inside the amygdala, there are cells that consistently get activated by the novel flavor. So these are what they call the novel flavor preferring neurons. So there are maybe 30% of the cells consistently get activated by the novel flavor. And then a different group of cells, maybe about 10, 15% of them, consistently get activated by water. Now, we don't know whether it is because these cells sense water or whether they sense the familiarity of water. These are kind of confounded because water is all by itself familiar. And then a third population of cells are classified as being non-selective. So either they don't respond or they respond equally to both the novel flavor and water. So using this neuropixel recording, they can divide the cells into either novel flavor preferring cells, population of cells, water preferring population of cells, or non-selective cells. So the question is, what happens when you put the mouse in a 30-minute delay, and then later on, you re-expose the mouse to the malaise? And in this case, they induce the malaise using optogenetic stimulation of the calcineutin gene-related peptide neurons in the parabrachial nucleus. So they artificially, using light, triggered the nausea in the brainstem. So what happened during the delay? This is very important and interesting because in the first hypothesis that we talked about, we proposed one of the way to bridge the gap in the delay, in the 30-minute delay, is that the flavor preferring neurons would persistently fire during this delay and bridging the gap. Did the researchers find that? Nope. During the delay, all the neurons in the amygdala basically stop firing. And it, in a way, maybe it makes sense because there's not much happening during the delay. But what happens during the light-triggered malaise? So now, during the stimulation period, when the researchers artificially activated the calcitonin-related peptide neurons, which we know from previous experiments we talked about, would trigger perhaps a sensation of malaise that would induce learning. What happens when they activated these nausea-related neurons is that in the amygdala, it preferentially reactivated the cells that were novel flavor preferring. So whereas when you pay attention to just the water preferring cells, the reactivation is very limited or non-existent. And also the non-selective cells didn't really care about anything. And you can actually zoom in and you can see that the reactivation of the flavor preferring cell is actually time locked to individual pulses of light. So it tells you that it is, and the researchers insightfully pointed out, this tells you that the reactivation is likely triggered by the direct kind of neural connection. It's a glutamatergic connection, so it's glutamate release. Direct glutamate release from these calcitonin peptide stimulation, and not by the alternative. An alternative is that the artificial light stimulation might trigger a generalized internal state of, oh, I feel sick, I don't feel good. But because this is so tightly time locked to the light stimulation, the researchers think it is actually a direct kind of connection that is triggering this firing, not due to kind of a global internal state. And interestingly, the researchers also found, so this is kind of, you can say this is kind of artificial. You're using light to trigger this malaise sensation. What about if you do lithium chloride, kind of more natural way of triggering this malaise? And the researchers found that yes, they found it basically the same thing when you give lithium chloride, in that the original novel flavor preferring neurons in the amygdala gets reactivated by lithium chloride, but not the water preferring neurons or the non-selective neurons. And it's quite
Tim, if you just didn't let them drink anything and just gave them lithium chloride, I presume these posterior neurons would fire, but what would happen in the amygdala?
That is a very interesting question that they don't talk about very much in this paper. They have one figure in the supplements where they just did a lithium chloride stimulation with a mouse that has presumably never seen Kool-Aid before, never seen the novel flavor before. And it doesn't look like it did very much, actually. Like, it doesn't look like it activated... Actually, let me take it back. It activated a subpopulation of cells in the amygdala.
In the amygdala? Really? Wow.
In the amygdala. But it seems like it's less than the 30% that they found with a novel flavor. But that's actually an interesting point. They didn't get into it. You would presume it would activate some cells in the amygdala that previously maybe the mice didn't like. Maybe they ate something that made them felt nauseous before, and maybe that would reactivate it. But they don't talk about it. But we might have time to talk about it a little bit more in the discussion when we can talk about some of the control that they could have done. So in summary, what researchers found is that the activation of the malaise sensation or artificial stimulation of malaise using optogenetics can reactivate the novel flavor preferring neurons in the amygdala. And it is relatively selective. It activates the flavor preferring neurons, but not the water preferring neurons. And they also, in addition, the researchers did some machine learning decoding analysis to show that you can basically train a decoder to decode the amygdala to classify whether it is when the amygdala the decoder would look at the amygdala population neuronal activity, and you can train it to say whether the mouse is licking the novel flavor or the water flavor. You train it, and then you put the same decoder during the malaise stimulation, the optogenic stimulation that makes the mouse feel sick, and ask what is going on in the amygdala. And the decoder says, oh, you are reactivating the novel flavor in the amygdala. So that tells you that the entire population is also yelling out novel flavor during the reactivation period. And they also did some fancy PCA, principal component analysis. So basically you can dimensionally reduce the 140 neurons that we record into just 2 dimensions. And you can ask a question, as the mouse is licking the novel flavor, how does the trajectory of neuronal firing, how does that kind of evolve in this dimensionally reduced plane? And what you see with during the exposure phase, the initial consumption phase, is that when the mouse is licking the water, the neuronal firing trajectory goes one way, but when the mouse is licking the novel flavor, it goes in a completely orthogonal way. It tells you that the amygdala can basically tell apart water and the novel flavor, which is what we found, because we found 2 different population of neurons. One prefer water, the other prefer novel flavor.
And when the amygdala fires in response to the flavor, the hindbrain region does not. The parabrachial nucleus does not, right?
Presumably not. They didn't directly record it, but based on the FOSS analysis from way back when we talked about the fMRI analog, the FOSS analysis, I don't think the parabrachial got activated during the initial flavor, which would make sense because it wasn't causing any nausea back then.
So basically what you have here is a default where you have this receptors for the nausea, the malaise in the parabrachial, and whenever they get something, they fire, and if there's some memory in the amygdala, then it'll have an effect.
Exactly. Presumably only if there's memory in the amygdala, it would trigger the reactivation of the amygdala. Yeah. And it's also shown in the trajectory when you stimulate the parabrachial nucleus, the neuronal firing of the amygdala as a whole looks like the trajectory of the firing looks like as if it's the mouse is experiencing the novel flavor. So all these come together to tell you, to suggest that, one important thing is in the group, if you record from the amygdala and you present lithium chloride as a way of reactivating the amygdala, this reactivation can be blocked by this reactivation of the amygdala population can be blocked if you ablate the parabrachial nucleus, the calcitonin peptide neurons in the parabrachial nucleus. So that tells you what goes on is that your stomach or your peripheral organs sends the lithium chloride that talks to the parabrachial nucleus, specifically the calcitonin related peptide neurons in the parabrachial nucleus, and that talks to the amygdala. So that if you delete those calcitonin neurons, then the malaise signal no longer can get through to the amygdala, and you cannot trigger the reactivation of the amygdala. So all this is coming together to tell you that the likely hypothesis that explains all this is the hypothesis where you have a specific population of neurons in the amygdala that gets activated by a novel flavor. Then during the delay, the activity goes away. But somehow, when you re-expose, when you are exposed to the malaise or to the optostimulation of the calcitonin neurons that triggers the malaise, the same group of neurons in the amygdala gets reactivated, and that somehow is important for the memory. So this is actually quite a very interesting point, because this has previously been seen before in the hippocampus in a process known as replay. So in the hippocampus, if you put a mouse in a maze and the mouse has to learn to turn left, turn right in a box, the hippocampus have these cells that are called play cells. So these are cells in the hippocampus that only fire when the mouse is in a particular place inside the maze. So people have talked of it as maybe part of the quote unquote GPS system of the brain. It tells you where you are in the environment. And if the mouse has been learning to do some task inside this maze so that it has to remember where it is to earn some food, then what happens is if you record from the hippocampus, as the mouse is learning, the hippocampus will fire in a very particular pattern based on where the mouse is, because it's going through different places. Your GPS is firing in a particular sequence. Later on, as the mouse is asleep, the same sequence gets replayed. And people think this is very important for formation of memory. So maybe if it's similar to like you sometimes can dream about what you saw during the day, maybe this is something quite analogous. Like when you are consolidating the memory, you have to replay what was happening when you're experiencing it. And this is a process that seems to be taking place in the amygdala, but no one has ever seen it before previously with taste aversion. So that is a novel finding that is quite interesting. And what's even more interesting is that this replay is not triggered spontaneously, but instead is triggered by the sickness, the malaise stimulus. Based on what we've shown so far, it seems that the malaise is very important in reactivating this flavor preferring neurons to fire during the malaise. But it doesn't really explain how the memory is, first of all, it doesn't really explain how the memory is formed and how it's retrieved. So in order to
also how it makes the mouse avoid drinking, right?
Exactly. In the future, how does the mouse actually remember?
Presumably the mouse has to sample before it can reject, right?
Exactly. It does. It does.
So how the hindbrain induced firing of the amygdala causes that, we don't know.
We don't know, and that's what we're going to talk about next. That's what the researchers address next. So to look into that, they extended the time. So right now, during the neuropixel recording in the amygdala, we focus on the initial consumption and also during the malaise phase, these two phases. The last missing piece of the puzzle is what happens during retrieval. So these researchers then, in order to compare all that, they looked at what happens during retrieval day when you expose the same flavor that the mouse now will reject to the mouse. So what they found basically was, if you look at the amygdala population as a whole, when you give the mouse the flavor that it now rejects because it's been paired with either lithium chloride or the calcitonin-related peptide neuron optostimulation, so the artificial firing of the neurons that makes the malaise. If you re-expose to the mouse on retrieval day to the same flavor, the same flavor-preferring neurons, the same cells, fire again. So this is a very stable population of cells that gets triggered during both the malaise, during the initial exposure, the malaise, and also the re-exposure. It's a very stable group of cells, and they saw it both with the malaise being triggered by lithium chloride and also by optostimulation. Okay, that's fair enough. That doesn't quite tell you how the memory is retrieved, because to really get to how the memory is formed, we should actually look at the other group of the other condition that we've kind of neglected up till now, which is the mice that got, instead of the Kool-Aid, the initial flavor being a novel stimulus, the group of mice, just remind our listener, the second group of mice, if they got the Kool-Aid as a familiar stimulus, if it's the fourth exposure of Kool-Aid, these mice actually learned that the Kool-Aid is maybe safe, and the malaise no longer triggers the learning to cause the aversion. So we know that a familiar flavor would actually not support this kind of flavor aversion conditioning.
Is that because the familiar flavor doesn't, the lithium after the familiar flavor doesn't activate the brachial neurons?
Exactly. So what they actually found, to look at that, they did the same amygdala recording, but in this group of mouse, they just exposed the mouse to the Kool-Aid, both initially, so when the Kool-Aid is novel, and also subsequently over several days, so as the Kool-Aid becomes familiar. And what they found is, during novel, when you present the Kool-Aid as a novel stimulus to the mouse, a bunch of amygdala neurons fire to the Kool-Aid, and it's a separate population of neurons relative to water. So this basically replicates what they found already. So you find your novel population of neurons that are sensitive to novel Kool-Aid, but over subsequent days, what they found was that this population of neurons, the firing basically goes down massively across days as the mouse becomes more and more familiar with Kool-Aid. So it's almost as if it goes from a novel population to a non-novel population. And in fact, when they use the principal component analysis, what they found is that the trajectory of the neuronal population of this novel flavor preferring population, on day one, they look very different from water. But by day 2 and 3, as they become familiar, these neuronal firing, the amygdala, basically looks more and more like if the mouse is just drinking water. Now, it doesn't mean that the mouse thinks Kool-Aid tastes like water, but it probably means the mouse is thinking the Kool-Aid is as safe to drink as water as a flavor. And what this suggests to the researchers is that basically what happens is that in the amygdala is that if you first time you give a novel flavor, the amygdala population fires to encode this novelty, and as the flavor becomes more familiar, this novelty, maybe alarm sensing neurons, they kind of goes away and becomes more like water, unless 30 minutes after the initial pairing, you give lithium chloride or you give some sort of malaise. In that condition, the novelty encoding neurons, they get reactivated, and now there's a memory that gets formed. So that subsequent exposure to the same flavor now reactivate the same population of neurons. And instead of in the familiar case where they kind of goes away, now it gets persistently activated, and the mouse kind of rejects it because of that. So this tells you this, as the researchers say in the paper, the role of the malaise and of the calcitonin-related peptide neurons is to stabilize these novel flavor-preferring population in the amygdala so that they fire persistently in the future. And this is the memory. The memory is that when you're re-exposed to the same flavor, the hippocampus, sorry, not hippocampus, wrong place, the amygdala, this specific population of cell refire again. So that tells you a little bit about how the amygdala is both forming the memory of the malaise, the novel flavor malaise, and also how it's recalling it. Mm-hmm. But the very last thing that the researchers addressed was still, we haven't quite answered during the 30 minutes delay between the novel stimulus, novel flavor, and the malaise. When these cells no longer fire, once again, remind our listeners, during the delay period, these cells, these population of cells, stop firing. So how come when you later on present the animal with a malaise, how come these cells know to refire? So very briefly, because we're running towards the end of the podcast, it is previously known that there's actually biochemically a very important important set of pathway inside the cell that are very important for plasticity formation. And they are the protein kinase A pathway, which involves a protein called protein kinase A. And protein kinase A basically it's a kinase, so it phosphorylates protein, and it phosphorylates this other protein called CREB. This is cyclic AMP response element binding protein. Doesn't matter, it's another alphabet soup. But this CREB protein is very important in basically both triggering synaptic plasticity, but it also can make the cell more excitable, more responsive. The cell can become more responsive to future glutamate input from another cell. And this effect can, and also another thing that PKA can do, and it can trigger FOS activation. So it all kind of goes together with what we've talked about so far. But by the proposal is that by activating PKA, you can make the cell more excitable for at least 30 minutes, maybe even longer, so that after 30 minutes, when the malaise signal comes in from the parabrachial nucleus, the cells, the specific cells that are activated by the novel stimulus, because there's this increased PKA and increased excitability, they would respond more. And this stabilizes this population of cell into what people call a memory engram. So to show this, the researchers basically did fiber photometry again. They expressed a dye, a calcium, back off, not calcium. They expressed a GFP protein that glows more when PKA is active. So basically, it's a GFP protein that is slightly misformed and has a different bit of protein that's stuck to it that can be phosphorylated by PKA. So when PKA is active and is phosphorylating, this secondary protein gets phosphorylated, and that reforms the GFP so that it now glows. So this is a way of basically telling you if PKA is active inside a cell. So when they did that, what they found is that when you present the mouse, well, once again, the question is, how does a novel flavor persistently stay active for 30 minutes until the malaise? So what they did was they presented this very simple experiment. They just presented a novel flavor to the mouse and see, does the amygdala neurons activate the PKA for, and if so, how long does it activate for? And what they found was, yes, the novel flavor, the Kool-Aid, when it's novel, it actually did activate the PKA. Two things are interesting with this finding. One is by day 2 and 3, so as the Kool-Aid become more and more familiar, this PKA activity is no longer seen. So it goes away very quickly. It goes away as the familiarity builds. And the second thing that's interesting is that this PKA activity goes on a little bit persistent. It stays on for about 20 seconds. So it doesn't stay on for the whole 30 minutes that we kind of thought might be required for the eligibility trace or the memory trace to form. It stays on just for 20 seconds at a time, but it comes up every time the mouse is drinking the Kool-Aid. And what is interesting with all this kind of secondary signaling pathway that PK is part of is that even though PK is active only for a little while, only for 20 seconds, the Krebs phosphorylation can go on for a long time because it's a persistent phosphorylation. And the Krebs activity can go on for even longer because it can act as a transcription factor to make genes turn on, and that can drive excitability. So that is what the researchers propose, is that it is this PKA that is only activated by the novel flavor is what is driving a potential excitability change in these neurons, so that 30 minutes later, when the Malay signal comes, they are more responsive. They haven't shown that these neurons are more excitable, but that's what they are proposing. So that's it. That is the whole paper. That's a lot went on, but just to summarize what the researchers found, they found, first of all, a mouse can bridge a 30-minute delay between Kool-Aid and subsequent sickness, and that the sickness signal is likely conveyed by the parabrachial nucleus, specifically these GCRP calcitonin gene-related peptide neurons. I think it's the first time I pronounced it correctly. They send input to convey this Malay signal, and they talk to the amygdala. And the amygdala is important both for the initial novelty, novel flavor induced neurons. So there's a population of neurons that encode the initial novel flavor. They get reactivated by 30 minutes later by the Malay signal, and they get further reactivated by the retrieval of the same flavor 2 days later as the mouse is rejecting it. And that the eligibility trace, the memory trace, is a big might. That more experiments need to be done, but it might be triggered, underlined, underlain, whatever. It's governed by the PKA. The PKA is what triggers these eligibility trace. So that's a summary.
So they say that our results reveal how dedicated novelty detection circuitry, so that's the amygdala, and built-in priors, preferential reactivation of recent flavor representation by post-ingestive malaise work together to enable the brain to correctly link stimuli and outcomes despite long delays. So I wonder if they could give a flavor and then 30 minutes, give lithium chloride, and then give a different flavor and 30 minutes later lithium chloride, and whether that would be able to distinguish the two. In theory, it would be, according to their ideas here. Right?
I think that would be a very poor mouse to have to endure 2 lithium chlorides. Yeah, wow. But I think that's actually a question. What is going on in the amygdala? Does it encode novelty, or does it encode something more specific to the flavor in addition to the novelty?
Yeah, because you're not going to encounter toxic stuff all that often, right? I suppose.
Hopefully not.
I don't know that you would need to, if you eat a bad meal, you're not going to have a bad meal for a while typically because you're sick.
Sure.
Right? So same with the mouse. So maybe the circuitry doesn't have to accompany more than one.
Right,
accommodate more than one, yeah.
Exactly. For your question, I have absolutely no idea. It's one of those things that the researchers, it would be very interesting for the researchers to look at.
This is great. It just raises more questions, which is the sign of a good
This is the whole
study,
point,
right?
Which is the whole point. I would say, relating to something that we discussed earlier, one thing that the researchers didn't quite do that would be amazing for them to do is if you make the novel flavor, the Kool-Aid, become familiar so that it no longer triggers a population firing inside the amygdala. Yeah. Then if you give lithium chloride, what does the lithium chloride do? This is something that you asked earlier, because now the amygdala presumably no longer has the memory trace, eligibility trace. You should no longer get reactivation, and therefore that's why you don't learn about a familiar flavor. Is that true? They didn't quite show that yet.
Yeah.
And another thing that's quite interesting is that during the very first bit of the talk, we talk about the lateral septum activating, preventing all this from happening. How does that work? We don't know. So the researchers also talk about in the discussion, like, does the lateral septum provide a safety signal? How does that all come into play? We don't quite know. There's a lot of fascinating science that we can follow up. And finally, this is in the amygdala, this memory lasts 30 minutes. How come we can't make the same thing happen for other kinds of learning? For example, tone shock pairing, which also happens in the amygdala. So what is so special about food and malaise? Evolutionary, it makes sense. Like food, malaise, this is something that I've probably hard-coded based on digestive tract links and stuff like that to be a very long, you have to be able to endure a very long delay. Whereas tone shock, usually nothing in nature is like 30 minutes before you hear thunder strike, and then 30 minutes later you get zapped by the lightning.
Well, I also make this statement that the major reason learning is challenging is because of delays between stimulus or action and outcomes. You can imagine you're studying for an exam, and then the exam is the next day or a week later, right?
Exactly.
That's a major delay.
There are two things. If you study for an exam, there are two delays you have to bridge. One is you have to remember the exam material today. Let's say your exam is in a week. You have to study today and remember the material in 1 week. That's one memory that you need to form. The second memory and delay that is even harder is I study the exam for now to pass the test next week. The reward is that maybe I would graduate or get a good job in 4 years. How do I bridge that delay? So there are delays on different levels in real life that an organism need to bridge, and we know very little about in neuroscience.
Well, in fact, people deal with that differently. Some people can't handle waiting 4 years, and they don't make it through college.
Exactly.
Right? Where others can just take exam after exam and move on, right?
Yeah.
It's just different wiring in some way.
Exactly. So what underlies all that? Is it because they can't bridge? Is it because the first group of people can't bridge the delay, or maybe they can't they don't know they don't learn about the delay, or they know about the delay but they just can't stand it? So there are a lot of questions that we just don't know quite yet how the brain kind of process all these things and come to some sort of judgment. But at least, sorry,
go ahead. At least
for the case of food. So the reason why this group of researchers, I think, try to address it using the flavor nausea paradigm is that it is a very evolutionary conserved paradigm, and the underlying circuitry probably is a little bit well worked out in terms of the parabrachial nucleus and even the amygdala. So they are able to kind of zoom in and kind of get a handle on this. So I think they've made some real progress, and it's very good to read this paper, and I definitely learned quite a bit.
Yeah, it's fascinating. I love it. This is the beauty of neuroscience, that it attempts to explain organisms' behavior, right?
Attempt being the functional word there, because it's very challenging. It can be quite challenging.
But we have great technology now, as you pointed out, that we didn't have years ago. Which allow us to identify neurons that are responding, right? For example.
That's right. So, but it's still constantly a challenge to make sure, like the experimental design, that you're isolating really the only thing that you're looking at, because there's a lot of things in the environment that can confound things, that can make things difficult.
It's Schrodinger's cat, right?
Exactly.
But it's beautiful because people love to understand why they act a certain way or react a certain way. And neuroscience does that. I mean, I would say virology also does it, because if you want to know what's going on when you're infected, immunology and virology will explain it too. But people are more enamored of neuroscience, it seems.
Well, related to neurology, this is basically 30 minutes from two-bucket disease triggered by a virus. So this is also important. Although I don't know how this kind of neuroscience I don't know how by a population of organism avoiding a food, how that kind of alters the evolution of these gastric viruses. Who knows? I'm sure it will have some sort of implication.
It's interesting because the virus could be present in different kinds of food. So if you eat potato salad at a picnic, right, and then you end up with norovirus gastroenteritis, I think you would eat potato salad again, probably, because you would know, that batch was contaminated. It's not a
right.
fundamental property of potato salad.
It's a familiar flavor. You've eaten potato salad before.
That's right. So, that's exactly what they find here, right?
But what's very interesting, like in terms of evolution point of view, is for an individual organism's point of view, it makes sense that you trigger this malaise and you throw up because you want to get rid of the poison before it does more damage.
Yes.
From a virus point of view, it also makes sense to make the organism throw up so that you spread it more, especially if you have, on a cruise ship or whatever.
So that makes it you wonder why mice haven't developed a vomit
exactly
reflex,
so
right?
Is it because they live in close quarters and they don't want to spread it? Is Maybe. It because their vomit is going to signal to predator that there's some mouse here and you can come eat them, although you might get sick after eating that mouse? Who knows? Yeah, we just don't know.
Yep. Anyway, thank you, Tim. That was great. Loved it.
Thanks, Vincent.
That's TWiN number 62. You can find the show notes at microbe.tv/TWiN. If you have any questions or comments, you can send them to twin@microbe.tv. And if you enjoy these programs where we talk about science, we'd love to have your support. We don't do advertising, as you notice. We just depend on listener donations. And you can go over to microbe.tv/contribute. Find out ways where you could help us. Tim Chung is at New York University. Thank you, Tim.
Thanks, Vincent. Yeah, it was fun and very nice paper.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.